Hi guys. Okay, so this round we're going to talk about carbohydrates and lipids. Let me share my screen. So we're going to start off with carbohydrates. This is the one most people are familiar with in general. They always call them carbs. And this is the one that is not as horrible and evil as people think. As a matter of fact, it's actually kind of necessary. So when we're thinking about our carbohydrates, this is going to be sugars and polymers of sugars. So sugars and things that are made out of sugar. Remember we said that polymers are when we take several individual units and put them all together into like a bigger molecule. So that's sugars and polymers of sugars. So one of the characteristics we're going to find with carbohydrates is that they will contain a carbonyl group, right? And many hydroxyl groups. So you'll notice we have lots and lots of the OHs, and we have a C double bonded to an O. Okay. They are made up of carbon, hydrogen, and oxygen. And typically we refer to the ratio, we say it's a 1 to 2 to 1 ratio. 1 carbon, 2 hydrogens, 1 oxygen. The reality is that as we build the polymer, that gets off because we keep pulling out the water, which means we keep losing 2 hydrogens and an oxygen each time we join them together. But if you account for that, then it still stays with that 1 to 2 to 1 ratio. So we call the simplest sugars, the monomer, the individual piece, a monosaccharide. Mono means one. So this is the monomer. This is what we build all the bigger carbohydrates out of, is the monosaccharides. The molecular formula has multiple variations. So remember, we said it's a 1 to 2 to 1 ratio, 1 carbon, 2 hydrogens, 1 oxygen. The most common monosaccharide is glucose. And so that's what we're showing examples of. Glucose is a nutrient and it is fuel for the cells. Most of your cells run specifically on glucose. Even if we get other molecules, we convert them to glucose typically to utilize to provide energy or fuel. Some parts of your body will not function on anything except glucose. So we use glucose in cellular respiration. We break it down and we turn it into the energy that the actual cells and body will use. Carbohydrates can serve as building blocks for amino acids or as monomers for di and polysaccharides. So remember, monosaccharides are the monomer, and so they're going to be monomers for disaccharides, polysaccharides, and we also can use that base monosaccharide as a building block to convert it into an amino acid, which then we'll use to build proteins. And so we'll talk about those when we get to the proteins. Okay. So a disaccharide, just like a monosaccharide, was one sugar, mono, one, saccharide is sugar. A disaccharide is two monosaccharides or two individual sugars joined together by covalent bonds. And remember, we use a dehydration reaction, dehydration synthesis, a condensation reaction. All three names are talking about the same thing. We use one of those reactions to remove water and join them together. The most common disaccharide is sucrose. If you go to the grocery store and you buy a bag of the white sugar, you are buying a bag of sucrose. Okay. The monomers, the two individual parts that make up the sucrose, are glucose and fructose. So we've put a glucose and a fructose together to make sucrose. Plants transfer carbohydrates from the roots to the leaves in the form of sucrose. So if you think about it, where we get our sucrose is from plants. We have some that's cane sugar. It's made from sugar cane. Another really common source is what we call beet sugar. You wouldn't know it's still sucrose, but if you look carefully, sometimes on the packaging, you'll see that it's beet sugar instead of cane sugar. And it's just from a different plant source. And then if we put many different monosaccharides or many different sugars together, joined via dehydration reactions, we get polysaccharides. Poly, many, saccharides is sugar, so polysaccharides. These are when people talk about complex carbohydrates, things like that, this is what they're typically talking about. And carbohydrates, when we're talking about polysaccharides, we're really talking about storage polysaccharides most of the time. So plants typically store their monomers of glucose as starch. So we just take glucose and join it with glucose and glucose and glucose and put a whole lot of glucose together, and we make these starch molecules. If you eat a potato, that's one of the key storage areas in a plant for this starch. Hence, potatoes are really starchy. This is a way that plants can store the excess glucose they make through photosynthesis for later use. Remember, plants make things for themselves. They don't make them for us. They make glucose and starches and things for themselves. We just utilize them, as do other animals. Animals store glycogen, which is also a polymer of glucose. So we can't store our excess glucose as starch like plants do. We store it as glycogen. So if you were, say, getting ready to have a big game the next day, sometimes the coaches or people who are advising you will talk about you need to carbo load. They want you to go eat a whole bunch of pasta, something like that. The idea is you're putting a lot of these carbohydrates into your system, and your body will store all that excess as glycogen. And the reason we like to do this is this can be broken down quickly and easily to provide your cells with fuel so that you can do those activities and have plenty of energy. That's why they often will suggest you do something like this. So glycogen is typically stored in the liver and in your muscle cells. Obviously storing in the muscle cells because that's where a lot of your movement and activity and need occurs when you need quick energy. And then there are also some larger polysaccharides that provide structural aspects. So cellulose is the tough substance that forms plant cell walls. Remember, plants have cell walls. We do not have cell walls. Okay? We cannot break down cellulose. There are some animals that can. Think about cows and such, and they typically have specific systems within their digestive system that are designed to help break down that cellulose. And then chitin, yes, it's pronounced chitin, not chitin. Chitin forms exoskeletons of arthropods, and we find a few other things that we find this kind of tough material that's chitin. It is also another polysaccharide, and these are structural, and once again, this is also something that we physically cannot break down if we eat them. So those people who eat bugs, they're breaking through the exoskeleton, and then they can digest and process the material inside the exoskeleton, but they can't actually process the exoskeleton. And I know some of you are going, ooh, eat bugs, but there's a lot of people in this world that is a main staple in their diet because it's protein that's available. And in reality, if you were ever put in a position where that's your option, you would probably eat bugs rather than starve to death. Okay, so practice. You are given an unknown monosaccharide. Remember, mono, one, saccharide, sugar, so one unknown single sugar monosaccharide to identify in the lab. The only clue you are given is that it has 4 carbons. You, being an excellent AP Bio student, figure out the formula and name immediately. What is the unknown monosaccharide? So I would start with the formula, and I would think about the ratio rule for our carbohydrates that we talked about. And then from there, you can work to identify the name. Okay, so that would be C4H8O4, 1 to 2 to 1, and it is known as threose. Have you noticed yet that all of these sugars, glucose, fructose, all of these sugars we keep seeing the names of end in OSE? That's a little trick you might want to think about. If you see a name of something that ends in an OSE, that's a very good sign that this is some type of sugar. And you're going to see that as we go through different molecules and things, that there sometimes are naming trends. So if you see OSE at the end, that's a really good clue that that's a sugar. Okay, now we have lipids. Lipids are a class of molecules that do not include true polymers. So it's not really the same piece put together string by string by string by string. But it still falls into the idea of macromolecules because we're using the hydrocarbon base skeleton and we're putting functional groups and enlarging it and putting large pieces together to form the final product. Okay. So in general, lipids are typically a fairly small size, especially when you're comparing them to our carbohydrates, our proteins, our nucleic acids, some of these other things. Some people don't consider them to be macromolecules because they don't fall into that true polymer concept, but for our purposes, and you will find that most general biologists do classify lipids within the macromolecule grouping. So lipids are considered nonpolar hydrophobic. This is the reason that, for example, when you pour oil into water, it doesn't mix because it's nonpolar. And remember, we talked about water and it's polar. And so they are hydrophobic, hydro-water phobic, fearing. So they don't like or they fear, they try to stay away from water. So the key types of lipids that we care about are fats, phospholipids, which play a really important role in terms of the cell membrane, steroids, which are a type of hormone, and yes, you have naturally occurring steroids in your body all the time. When people talk about steroids in terms of doping for sports and things like that, they're talking about artificial or added injected steroids, not the naturally occurring in your body steroids. So we're going to start off with fats. Fats are composed of 2 main portions, a glycerol molecule and 1 or more fatty acids. Glycerol would be classified as an alcohol, and a lot of alcohols you'll see end in -ol, ethanol, isopropyl alcohol, but the OL is one of the key things you'll see in a lot of the alcohols. Okay. So glycerol is what we consider an alcohol because of the hydroxyl groups. And then fatty acids are long carbon chains with a carboxyl group at one end. And so we may have 34 carbons joined together in one single chain. They're really long carbon chains with hydrogens on them. So the most common fat that we deal with is what we call a triglyceride. We have tri is three fatty acids joined to a glycerol via what we call ester linkages. Really and honestly, what we're saying is we use dehydration synthesis to join the pieces together, but because they're in fats, we call them ester linkages. So you're going to find that we use that same dehydration reaction and hydrolysis for all of these molecules that we're going to talk about, but depending on the molecule, they call the bonds different bonds. So these bonds between the fatty acids and the glycerol, we call ester linkages. So the bond will occur between a hydroxyl on the glycerol and a carboxyl group on the fatty acid. Okay. So we classify our fats as either saturated fatty acids or unsaturated fatty acids. And this is something you've probably heard about when they talk about saturated fats and unsaturated fats. But did you really understand what they were telling you? So a saturated fatty acid has absolutely no double bonds between the carbons in the carbon chain. So that means we have more hydrogens. It's saturated with hydrogen. These tend to be very straight chains. So in reality, if you think about it, if they're really straight chains, then we could stack one on top of another, on top of another, on top of another, on top of another. They'd stack very well. An unsaturated fatty acid is going to have one or more double bonds. And if you remember back, we talked about the fact that double bonds typically cause it to bend or kink. So instead of being straight, it's going to twist or kink. And so if you have these bends or kinks, they're not going to stack up as neatly. So they tend to be more liquidy, whereas saturated tend to be more solid. Okay, moving on to the phospholipids. And remember, like I said, phospholipids are the key component to a cell membrane. And so we really need to understand these. And a phospholipid has two key parts, as you can see in the diagram. We have what we call a hydrophilic head and a hydrophobic tails. Hydrophilic, hydro water, philic loving. So hydrophilic head kind of likes water. And the hydrophobic tails, we know about hydrophobic and they don't really like water. Okay, so these are the major component of cell membranes. We have two fatty acids, so that's what the tails are, attached to a glycerol, and that glycerol has a phosphate group on it. So that's what makes the head is the glycerol and the phosphate, and then the tails would be the fatty acids. When they are placed in water, they actually naturally assemble as a bilayer. So what happens is the tails will get hidden in the middle and the heads will face outward, and it forms like a little ring so that we're protecting the heads. So let's see if I can. So we've got the heads and the tails, right? And so they're literally going to face each other, right? So what happens is we make a ring that has the two sets. So we have a ring that goes for facing out, and we have a ring that goes for facing in, and it makes a little bilayer or circle. Kind of think of a donut. We got the inside of the donut and we got the outside of the donut. Okay. So the tails are hydrophobic. Those are the fatty acids. The heads are hydrophilic. They like water, so that's the glycerol and the phosphate. Okay. And then we have steroids. You'll notice steroids have a ring structure. Typically, they are based off of a 4-fused ring structure. Different steroids are going to vary, but they're all based off of the same structural molecule. So unique groups attach to the rings to determine the type of steroid. For example, this one is testosterone, which is one of the key hormones in males. So that kind of gives you some background and some information about the carbohydrates and lipids. Hopefully that helps. And we'll be talking about proteins in our next video.